I'm Muzzleson.
And I'm Stahl.
And I was born May 24th, 1930.
I was born October 8th, 1929.
And this is how our experiment worked. First, we grew DNA in an N15 environment. N15 is an isotope of N14, and it is much denser. My colleague and I were thus allowed to use density labeling techniques to track the DNA. We then placed the bacteria in an N14 environment. We then placed the bacteria in a cesium chloride solution and in a centrifuge, allowing a concentration gradient to form. My colleague and I then observed a phenomenon. An intermediate type of DNA had formed. We thus concluded that DNA is a semi-conservative process, as one strand of the N15 DNA had been joined with one strand of the N14 DNA. Thank you. First, helicase comes in, separating the DNA along with single-stranded DNA-binding proteins to prevent the DNA from reassociating. When DNA replication occurs, it must occur in the 5' to 3' end. This occurs effectively in the leading strand of DNA. However, the lagging strand presents problems. The DNA can only be produced in fragments, which are separated by RNA primase. These fragments are called Okazaki fragments. Then DNA polymerase I comes in and lays down the DNA where the RNA primase I once was, and finally DNA ligase comes in and links the Okazaki